All right, you guys, let's go ahead and start. So today we're doing exam review for exam 4. And remember, this exam covers chapters 8, 10, 11. All right, so let's go ahead and start. Chapter 8 covers photosynthesis. We should obviously know what photosynthesis is, which is the process of using that solar energy, converting that into a chemical energy that we can use, right? Sugars and things. And directly or indirectly, this nourishes all of life. You should know what autotroph means. These are organisms that can sustain themselves, usually through photosynthesis. And you should know what kinds of organisms do photosynthesis. These are the ones listed here. You should know what a heterotroph is. For example, humans are heterotrophs. We cannot sustain ourselves. We need to eat. We need to get our nourishment from something else. We can't just go out in the sun. Get sunburned. You should know the structure of a plant, or leaf, I should say. Remember, it's the mesophyll cells, the cells in the middle of the leaf, that contain the chloroplasts, right? So you should know the mesophyll cells contain the chloroplasts. And you should know that the stomata, see at the top right there, the stomata are little pores that open and close that allow gas exchange to happen. CO2 comes in mostly, and O2 comes out. That's a net O2 release and CO2 uptake. And then you should know that the chloroplast has this structure here. Outer membrane, inner membrane, stroma. You should know the terms granum, thylakoid membrane, thylakoid. Thylakoid space is the space inside. All of that stuff is good to know. You should know the equation for photosynthesis. That, you know, what are the reactants? The reactants are CO2 and water with the help of light energy. And then what are the products? The products are glucose and oxygen. Right. And you should know what's becoming reduced. The CO2 gets reduced. It gains electrons. The CO2 gets reduced to sugar, whereas the H2O gets oxidized. Remember, H2O is the electron donor. In fact, you should remember that H2O is the primary electron donor, right? It's the first electron donor. It's where the electrons come from, right? What else? We talked about the 2 stages of photosynthesis. Remember the light reactions followed by Calvin cycle. You should know the ins and the outs of all of those, right? The light reactions, water and light are reactants, are used. Oxygen is a waste product. ATP and NADPH are useful products, right? And remember, all of the light reactions happen in the thylakoid, specifically the thylakoid membrane is where everything's really going on, right? Remember that? And then the Calvin cycle happens outside of those thylakoid pancake stacks, I call them, right? The Calvin cycle happens in the stroma. And you should know, this requires CO2 from the air with carbon fixation, and it requires the ATP from the light reactions. By the way, this ATP was made by photophosphorylation, not oxidative phosphorylation, not substrate level phosphorylation. Photophosphorylation is how this ATP was considered to be made, right? NADPH is also required. ATP is required for Calvin cycle. CO2 from the air, and the product is sugars, right? Then we talked about light, and light consists of electromagnetic radiation with different wavelengths. You've got this radiation of different wavelengths. All the wavelengths together make white light, and then you break them up into different wavelengths, and those represent the colors, right? You don't need to memorize that 380 is purple and 600 is yellow. Don't memorize that. But what should you know? You should know that short wavelength means high energy. Long wavelength is low energy. Yeah. And what else do you need to know? You need to know about the pigment molecules. Remember, because these pigment molecules allow photosynthesis to happen. The pigments are the chlorophyll A, the chlorophyll B. You know, other pigments exist as well, like xanthophyll and carotenoids. You don't need to know the xanthophyll and the carotenoids, but you know, pigment molecules are required for using that sunlight energy, capturing that sunlight energy. And remember, do the pigments absorb green light?
No, they bounce back.
No, that's right.
The color you see that mean the
Transmitted. Transmitted means the color goes right through, or reflected, which means bounces right off, right? So what you should know is green light is transmitted or reflected. Green light is not absorbed. Green light is not absorbed. Okay? That's a good thing to know. You don't need to know the difference between chlorophyll A or chlorophyll B. I have this here to show you that chlorophyll is an organic molecule. It has a cofactor, right? It's pretty interesting. I just wanted you to see what it looks like. I don't need you to memorize this. Just good to understand that, again, sunlight or any kind of visible light, it hits the chlorophyll molecule, it excites the chlorophyll molecule. That means its electrons go to an excited state. And when the electrons go back to normal state, it releases a bunch of energy, right? And then we talked about the photosystems. Remember, the light reactions of photosynthesis depend on the photosystems. Then the photosystems have two main parts, right? The purple part is called the light harvesting complex. That's where it starts, right? That's where the chlorophyll and other pigments live, right? When light hits a pigment, that pigment becomes excited. Its electrons go to a higher energy state. When the electron comes back to its normal energy state, that pigment releases energy, which excites its neighbor. That neighbor is now excited. That neighbor can excite its neighbor. And the excitement gets harvested. That's why it's called a light harvesting complex. It's harvesting light's excitement. It's harvesting light's energy. And that energy is bouncing around and reaching the central chlorophyll pair in the blue part, right? The blue part is called the reaction center. And it has two special, a pair of chlorophyll A. And remember, real quick, let me show you this. In photosystem 2, the pair is called P680. In photosystem 1, the pair is called P700. So again, just to show you what I'm talking about, this pair on photosystem 2 is called P680. This pair on photosystem 1 is called P700. That's what I'm talking about. They have names. The special pair have names. And remember what the special pair do when they get excited? They release the electrons. Not just allow the electrons to go to a higher energy state and then back to normal. They actually release those electrons. The electrons move to the primary electron acceptor, at which point the electrons can completely leave and abandon the photosystem. The electrons go on a journey. See this yellow arrow? The electrons are going on that journey now, right? The electrons go through an electron transport chain, which, by the way, you don't need to memorize the plastoquinone and cytochrome complex and PC. You don't need to memorize the name of it. Just know there's an electron transport chain. The electrons go through this electron transport chain, pumping protons into the thylakoid space. And the electrons do what? They get stuck here at the central chlorophyll pair on photosystem 1. They get stuck there for the time being. What happens when you have a lot of protons here in the thylakoid space? There's a high concentration. They want to diffuse out through chemiosmosis. They will diffuse out, but through that they will spin this motor, ATP synthase, that produces ATP in the stroma via photophosphorylation. Remember, photophosphorylation is the name of that. So that was the whole reason electrons go through this electron transport chain. That's powering the electron transport chain to pump protons, have a high concentration of protons. Those protons chemiosmose out. They exert that proton motive force, remember, spinning ATP synthase to make ATP with photophosphorylation. And remember, the electrons were stuck here at the central chlorophyll pair on photosystem 1, right? And they have to leave that, right? How do they leave? Again, sunlight has to hit a pigment in this light harvesting complex. That pigment gets excited, which can then excite its neighbor, which can then excite its neighbor, and the excitement reaches the central chlorophyll pair. Now those electrons are free to move on their journey and go to that enzyme called NADP+ reductase. That enzyme hands the electrons to NADP+ to make NADPH. So the electrons went on a journey from this end to the other. And where did the electrons originally come from? From H2O. So what happens is, what happens is this special pair, when light hits and excites the special pair releases electrons. This P680 releases electrons. Okay, and let me tell you about that real quick because we didn't go into too much detail about that. But look what's happening. See, this slide is showing you what's happening there, right? Light hit photosystem 2. That excited this pigment, which excited this pigment, which excited this pigment. The excitement reached the pair, the P680 pair. P680 released the electrons to the acceptor, and those electrons went on a journey, right? They went through the electron transport chain and whatnot. Now look what happens. This is slide 2. This P680, once it gives away those electrons, it becomes what's called P680 plus. Does that make sense? If you give off electrons, now you're plus charge, right? Make sense? Mm-hmm. It's called P680 plus. And P680 plus is a very strong oxidizing agent. It's super greedy for electrons, right? Does that make sense? And with the help of an enzyme, it can steal even from water, right? Which means you're stealing from oxygen. Isn't that super powerful? I mean, think about it. If you're stealing electrons from oxygen, you have to be highly oxidative, very greedy, right? Because oxygen's very greedy, and you're stealing from oxygen, right? So this P680 plus is actually even greedier than oxygen, which is wow. You know, that's like stealing from a master thief. You know what I mean? Like, you know what I mean? Like the thief stole a painting, and then you stole the painting from the thief. You know what I mean? Because like oxygen is the thief. It can take electrons and become water, but you're taking the electrons from the water. You know, it's amazing. So that's how strong the P680 is. When it loses its electrons, it becomes P680 plus. Suddenly it goes from, here, take my electrons, and then suddenly it becomes P680 plus, the most greedy thing in the world. You see, it's kind of like a switch. Does that make sense? Mm-hmm. When P680, I'm sorry, hold on. Listen to this. When P680 has electrons, and then the excitement of light reaches it, it goes, here, take my electrons. It has a very negative EO. You see what I'm saying? It doesn't care about the electrons. If P680 has electrons and light energy reaches P680, it switches to negative EO. Remember negative EO, negative reduction potential? Here, take my electrons. I don't care, right? But now what did it become? P680 plus. And it just switched, like it went from negative EO to super positive EO, right? Does that make sense? Like a switch, like a light switch, off, on, right? It went from negative EO and so positive it can steal from water. Does that make sense? So P680, it's considered a reduction potential switch. Does that make sense? Why? Because if P680 has electrons and the light energy reaches P680, it goes, meh, here, take my electrons, negative EO. But now it's called what? P680 plus. Whoa, switch to positive EO, positive reduction potential, super greedy, super duper greedy, right? I can steal from even water, right? So it's very interesting. It's very interesting little pair, isn't it? It can go from very negative EO, and once it gives away those electrons, very positive EO, right? And that's how it reloads itself. It reloads itself when it's P680 plus because it's so greedy, and then it gives away the electrons easily when the energy, light energy reaches it because it's not greedy. It's like a switch. Does that make sense? So that's good to understand, you know. Obviously, we didn't go into it too much during the lecture, but, you know, if you understand that, it might be beneficial, hint, hint, you know, for some extra stuff going on. I don't know. That's a good hint. But look at that. Yeah, it's all explained here on this slide.
Is it the same for the P700?
Well, actually, no. That's a good question. Because I'll explain why in a minute. The question was, is that the same thing that's going on with P700? And I'll explain, not quite, not as extreme, not as extreme. So What happens to the electrons now?
It goes to
They leave, right? They go through PQ, cytochrome complex, PC. Remember the electron transport chain? That pumped the protons, and that led to ATP production, right? Photophosphorylation, right? And then the electrons got stuck where? P700 on photosystem 1. Remember that? And they stuck there until, see that light over there? Until light excites this pigment in the light harvesting complex, which excites that pigment, which excites that pigment, which excites that pigment. And when the excitement reaches P680, now P700, it switches to
less.
negative EO. It lets go of those electrons. Now it's P700 plus. It is kind of greedy, but not as greedy as P680. It's not as extreme. Not
So
as
it cannot steal.
it can't steal from water. So P700 can't get reloaded directly from water. It's not that greedy,
Okay.
right? It is kind of a switch, but a weak switch, right?
That's
Does
why
that?
you cannot take water.
Yeah, it can't take it from water. Only P680 can take electrons from water. It's that greedy. P700 is not nearly greedy enough, right?
So when that system shut down now.
Yes, if this isn't working, that's not working. That's right. That's right. Okay. And remember, now P700 can release those electrons, and the electrons can leave. Where do they go? To that enzyme, NADP plus reductase, the enzyme which hands those electrons to NADP plus to make NADPH. So the electrons started on water. So you can call water the donor, the initial electron donor, or the primary electron donor, right? Primary electron donor.
Is it oxidate?
It gets oxidized. Yeah, water gets oxidized because you're stealing its electrons. And then those electrons go on a journey, and then NADP plus gets reduced to NADPH, right? Picks up the electrons at the end. NADP plus gets reduced to NADPH. And during the light reactions, just the light reactions, these are the light reactions, NADP plus is the terminal electron acceptor, right? It picks up the electrons at the end of the light reactions, right? I don't know if I asked that, but if there's a question like, what is the terminal electron acceptor during the light reactions? NADPH. But what's the terminal electron acceptor for all of photosynthesis?
Oxygen. CO2.
CO2. Because that CO2 is going to pick up the electrons to become sugar.
Sugar, yeah, glucose.
Right? Does that make sense? Okay, good. You don't need to worry about cyclic electron flow. I know we didn't have time to touch on it too much. Don't worry about that. What else? You should be able to do this, though. Here, this is actually something interesting. You should be able to compare and contrast the mitochondria and the chloroplast in terms of three things. Where's the proton concentration high in both? Where's the ETC and the ATP synthase in both? Where is the ATP being made in both? Right? And it tells you right here, the proton concentration high in the mitochondria, it's in the intermembrane space. Remember? That's where the proton concentration high. In the chloroplast, it's in the thylakoid space, inside each of those little pancakes, right? Where's the ETC and the ATP synthase in both? In the mitochondria, it's in the inner membrane. It's part of the inner membrane. It's in there, embedded. And in the chloroplast, it's in the thylakoid membrane, right? And where's the ATP being made? In the mitochondria. The ATP is actually being made in the matrix of the mitochondria, right? But in the chloroplast, it's being made in the stroma, right? And in the mitochondria, it's being made by oxidative phosphorylation. In the stroma, it's being made by photophosphorylation. Remember that term? Photophosphorylation. That's how you're making the ATP. Let me just check on this. Yep, it's still working. Great. All right, any questions so far about anything? Pretty straightforward stuff. Okay, so again, These are the light reactions. They depend on light. Obviously, without light, the electrons aren't going to flow. The waste product is oxygen, and the useful products are NADPH and ATP. Okay, not glucose here. Not glucose here, because these are just the light reactions. There's no glucose being made during the light reactions, right? Light is required. Water is required. Water is the primary electron donor. Right? And then we have the Calvin cycle, which, remember, there are 3 phases to the Calvin cycle. You should know the first phase is called carbon fixation, which is catalyzed by the enzyme rubisco. The enzyme is called rubisco. Then you have phase 2, which is reduction, which means giving the electrons to the CO2 to make sugar. And then the third thing you need to do, the third phase, regenerate the RUBP, the ribulose bisphosphate, that CO2 acceptor, right? So let me show you. This is the Calvin cycle. You don't need to know every single little step. But what do you need to know? You need to know the main things. The enzyme rubisco. I know it's weird because I told you before that enzymes usually end with ACE. ACE. ASE. This is an example of an enzyme that doesn't end in ACE. Okay, it's rubisco. Rubisco is an enzyme. It's the enzyme responsible for carbon fixation. What does that mean? In this case, it means taking CO2 from gas form and fixing it, joining it to the carbon dioxide acceptor. This organic molecule will accept the carbon dioxide, right? And that is called ribulose bisphosphate. The short for that is RUBP. Okay? So, what happens first? Phase 1, carbon fixation. The enzyme, rubisco, joins CO2 from gas form, from the air, to the CO2 acceptor, RUBP, to make, it doesn't matter what it's called when you make it, to fix the carbon dioxide. That's carbon fixation. You don't need to know what you make, the phosphoglycerate. You don't need to know that. Then what? Phase 2 is reduction. That's CO2. Now you have C's and O's, right? You're trying to make what? Sugar. Sugar is C's, H's, and O's in a 1 to 2 to 1 ratio, right? Do you have C's and O's? You captured C's and O's from the air. What else do you need to make sugar? H, right? H's, right? The H's are coming from where? NADP, H. That's what reduction means. I'm going to give these H's, which is a proton and an electron, I'm going to give those H's to the CO2 I captured. Now I have C's, H's, O's, and that takes energy to make sugar. It's not easy. That's why we need that ATP from the previous light reactions. Right? Does that make sense? So phase 2 is called reduction. You're reducing, you're giving electrons to, H's to in this case, the C's and the O's, right? To make C's, H's, and O's, which is sugars. Okay, glucose. And then what's the last thing you need to do, phase 3? Regenerate this guy, right? The CO2 acceptor. You have to regenerate the CO2 acceptor called ribulose bisphosphate, RUBP, so that you can do this again, right? Just like at the end of the at the end of the Krebs cycle. Remember the Krebs cycle? At the end of the Krebs cycle, you had to regenerate oxaloacetate. At the end of the Calvin cycle, phase 3 is regenerate ribulose bisphosphate, RUBP. It makes sense because it's going to accept the next CO2. You see what I'm saying? All right, questions about that? One more thing. So what is required for this? CO2 from the air, right? ATP and NADPH. And what is produced? Sugar. You could call it glucose, right? Even though the first thing is glyceraldehyde 3-phosphate, 2 3-carbon sugars, and then they join together to form glucose. You don't need to know that. Just know the main product here is glucose, right? Any questions? And where is this all happening? This stroma. Yeah, it's the fluid outside of the granum, outside of those stacks.
Granum's only light, right?
Yeah, the membrane of the granum is called the thylakoid membrane. That's where the light reactions are happening.
So the oxygen released in the light reaction?
Yes. Yes, and the CO2, the oxygen is released in the light reactions, and the CO2 is being used up in the Calvin cycle. Yep. And so CO2 is becoming reduced to sugar. And that's that. And that's chapter 8. If you understood those concepts, if you could follow that, you're going to do just fine on the exam. In chapter 10, I've already posted some nice videos on chapter 10. So if you are confused on any part of chapter 10, with the mitosis specifically, I have some videos breaking down each and every step, interphase and mitosis. But let's go through this just in case. So know that what the cell cycle means. Okay, what's cell division? Very straightforward. Know what the genome is. It's all of the genomic material, right? All of the DNA inside of the nucleus is your genome, and that's made up of chromosomes. Chromosomes are continuous pieces of chromatin, right? How many pieces of chromatin do humans have? How many chromosomes? 46. We have 46 chromosomes because we have 46 pieces of chromatin, right? You could think of it that way. And the chromosomes are very long. And different parts of the chromosome are called the genes. Those are the parts that code for the proteins, right? Genes are the information to make a protein. And each chromosome is very long. It has many genes on it, right? So somatic cells are your non-reproductive cells. Gametes are the reproductive cells. Mitosis results in somatic cells. Meiosis results in gametes, sperm and the egg. Somatic cells are diploid, which means two homologous sets of chromosomes. Gametes are haploid, one set of chromosomes.
Are germ
Yes.
cells heavily careful?
Ah,
Somatic, or
that's a good question. Germ cells. The germ cells are the specialized cells that become the sperm and the egg after division with meiosis. Germ cells start somatic, and then they become gametes. They're very special because they're the only cells in your body that do that. They're the only cells in your body that go from somatic to gamete because they start as diploid, but they end as haploid, right? And those are the germ cells. Those are the ones in your ovaries or the testes. Good question. So remember we talked about chromosomes. They're made out of chromatin. If the chromatin is loose and the genes can be expressed, it's called euchromatin. If the chromosomes are kind of condensed and the genes can't be read, that's called heterochromatin. You know, I don't know if I even asked that, but it's good to understand. What is a nucleosome? It's one unit of chromatin, right? Yeah. What is a nucleosome? One unit of chromatin. What does that mean? 8 histone proteins with DNA wrapped around, right? That's a nucleosome. And what's chromatin? A bunch of nucleosomes, right? Chromatin is many nucleosomes, right? And the DNA is loose like this, like a bowl of spaghetti. The DNA is decondensed during most of interphase, right? But then the DNA condenses into what's called mitotic chromosomes. That's the only time it looks like this, is when the DNA has fully condensed. The DNA only looks like this during mitosis. During mitosis, which is only remember, about 10% of the cell cycle, right? The other 90% of the cell cycle, the DNA doesn't actually look like this. It looks like that. It's just chromatin, like a bowl of spaghetti. Okay? What else? We should know the terms diploid and haploid. Diploid, they denote that as 2N. Haploid, N. Okay? N stands for set. Okay? Homologous set. By the way, you should edit your notes. This is actually very important that you understand. Diploid doesn't only mean 2 complete sets of chromosomes. It's 2 complete homologous sets of chromosomes. There's a big difference. Yeah, mom and dad. Right. So do you know what that means? You need a set from your mom, and you need a set from your dad. That's when you're diploid. You can't just take, for example, you can't just take a set for your mom, double everything, and now you have 2 sets and you're diploid. That's cheating. You need a set from your mom, 23 from your mom, and a set from your dad, 23 from your dad. So you have 2 chromosome 1s, but they're not the same. One's from mom, one's from dad. If you don't have that, 2 homologous sets, you're not diploid. And remember, that's the reason the two cells at the end of meiosis 1 are called haploid,
Even they have 46.
even though they have 46 chromosomes. Does that make sense? Because it's very confusing for students. They think, well, at the end of meiosis 1, I have 2 cells with 46 chromosomes, which means each cell has to have 2 sets of chromosomes, but why are we calling them haploid? Well, because you don't have a mom set and a dad set to back it up. You don't have a mom and a dad chromosome 1. You don't have a mom and a dad chromosome 2. You just have 2 of mom's chromosome 1. That's not enough, right? Does that make sense? So that's good to understand. We should know humans have 46 chromosomes, right? Their sperm and the egg have 23. Definitely know the difference between homologous chromosomes and sister chromatids. Homologous chromosomes are the paternal and maternal set of chromosomes. They are not identical. One was inherited from mom, one was inherited from your dad. Okay, they are not identical. When these get copied during, remember, S phase, only after S phase, not before, only after S phase do you have sister chromatids, right? Sister chromatids do not exist in the cell before S phase. Before S phase, you just have the homologous chromosomes. Then you copy everything. Now you've got 2 exact copies of dad's chromosome 1. These are sister chromatids. 2 exact copies of mom's chromosome 1, sister chromatids. So what you could say is this pair of sister chromatids is homologous to this pair of sister chromatids. What does that mean? For example, this is both of dad's chromosome 1. That's homologous to both of mom's chromosome 1, because this is the dad chromosome 1, this is the mom chromosome 1 copies, right? Does that make sense? And remember, you need to know the terminology here. Chromosomes, these are mitotic chromosomes. Remember when the DNA has really super condensed, which means it's mitosis time, right? When the DNA is condensed into mitotic chromosomes, it has a narrow waist. That's called the centromere, right? The sisters snap together with cohesin proteins. Yep. And on the outside, kinetochore, and that's responsible for microtubule attachment, right? Microtubule attachment. Yeah. And remember, the kinetochore, it's not drawn here, but remember the kinetochore has little motors inside. Motors. And that's why the sisters can move apart, right? During anaphase, they move. They're moving because there's a motor inside of the kinetochore. So the chromosomes themselves move, right, when anaphase happens. Good. We talked about the cell cycle. 2 parts of the cell cycle: M phase, which is only like 10% of the cell cycle; interphase, the other 90% of the cell cycle, roughly, right? M phase means mitosis and cytokinesis. Cytokinesis is not part of mitosis, but they're together, they're M phase, mitotic phase. Interphase has its own 3 subphases: G1, S, G2, right? Mitosis has its own subphases: prophase, prometaphase, metaphase, anaphase, telophase. And then remember, at the end of mitosis, you have cytokinesis starts. Oh, and by the way, again, cytokinesis is not part of mitosis, but there is overlap. They're concurrent. So what does that mean? Before telophase is finished, cytokinesis can start. But that does not mean cytokinesis is part of telophase, or cytokinesis is part. It just happens to overlap, right? Does that make sense? So let's break it down. Interphase. And again, remember, I have all these videos already, but this is just another review. These videos are on YouTube already. what happens during G1? The main thing, I'll just tell you the main things to know, okay? The main things to know. Obviously, there's more to know than what I'm saying, but these are maybe the main things to focus on for the test. G1, the cell grows, right? Oh, by the way, if you don't pass G1, what do you enter? G0, yeah, G0. If you never pass G1, that means you exit the cell cycle and enter G0. Remember that? S, DNA is synthesized, right? You copy all the DNA. Now you have sister chromatids. So you went from 46 chromosomes to 92, right? G2, the centrioles replicate. Now you have two centriole pairs, and they start moving apart. Good. Opposite poles. And notice again, do you see those little X-shaped chromosomes in the nucleus? Not yet. Not yet, because the DNA is starting to, at the end of G2, the DNA starts to condense, but it's not mitotic chromosome yet. It's not, yeah, it's not condensed yet, all right? Does that make sense? So it's not until prophase that the DNA condenses. Now you can see those X-shaped structures, and each of those structures is a sister chromatid pair. So that's the important thing to know about prophase. The most important thing to know about prophase for the test is sister chromatid pairs form. The nucleus is still there, isn't it? But it's starting to break down, but you should know the nucleus is still there. But it's starting to break down, right? Those centrioles are moving apart. Okay, and the mitotic spindles form. Yeah, mitotic spindles form. Prometaphase, the nucleus has broken apart, right? It basically breaks apart into little vesicles, right? And the sister chromatid pairs are kind of floating around. What is the main thing to know? Again, the nucleus breaks down, and microtubules from the centrioles, from the mitotic spindle, microtubules start attaching to sister chromatids at the kinetochore, right? And the sisters start making their way to the center of the cell. They're not quite there yet, okay? What else? Oh, one more thing you should know. The centrioles, we stopped calling them centrioles right about, we call them centrosomes, centrosome. What does centrosome mean? Centriole plus all of these spindle, microtubules, all that. So yeah, they stopped calling them centrioles because it's more than just centrioles. It's centrioles plus all these microtubules, right? So it's called, this is called the centrosome. This is called the centrosome. So that's where the microtubules are coming from, the centrosomes. Okay? Metaphase, what do we need to know? That the most important thing to know is what's lining up. The sister chromatid pairs are lining up down this imaginary plane called the metaphase plate. Right? It's an imaginary plane. Of course, it's not a real plane. Sister chromatid pairs line up. Right? Well, the cohesin proteins are still there until anaphase. The cohesin proteins let go, right? And the important thing to know about anaphase is what's separating from what? The sisters are separating from one another, right? The sister chromatid pairs, the sisters separate from one another, right? Which means one copy of the information is going this way and one copy of the same information is going the other way. And why are they moving? There's motors in the kinetochore. They're not being pulled by the centrosome. The centrosome is not pulling the chromosomes. The chromosomes are moving themselves. Yeah, because they have a little motor, and the motor walks along the tubule. Right.
So just the microtubule, just like the guideline, like the direction.
Exactly. Like imagine if there's a rope over there, and I'm holding the rope. I myself am pulling the rope and making my way over there.
So
Yes.
the chromosomes from the first cell and the chromosomes from the second cell, they should be identical.
Yes, yes. Yes, exactly. Because what you've essentially done in mitosis, and that's why the product of mitosis is two genetically identical cells. The reason for that is you started with 46 chromosomes. All you did was copy every single chromosome, make an identical copy. And then what you're doing now is sending one copy one way and another identical copy the other way. So you're going to make two identical genomes. The two daughter cells are going to be genetically identical. to each other and to the original cell that made it.
Okay.
Does that make sense?
And like meiosis is one
It's
of the.
different.
Oh, meiosis ends up with 4 genetically non-identical cells. So none of those 4 cells are genetically identical at all, right? Yeah, so telophase, the main thing to know, daughter nuclei form, right? Daughter nuclei form. And what have you achieved? At the end of mitosis, you've achieved 2 genetically identical cells. Good. Then we talked about cytokinesis. Remember, this happens differently it happens by cleavage furrow formation in animals. And the protein very responsible for that, or most responsible for that, are these actin microfilaments, remember? That's responsible for the cleavage furrow, most responsible. Of course, there's other proteins that play a role, but the main factor is the microfilaments. That's responsible. In the plant, you can't do that. In the plant, we have cell plate forms. That's what we need to know. A cell plate forms, and that matures into a new cell wall. And that's how you do cytokinesis in a plant. And by the way, I don't know if I asked this, but these vesicles full of cell wall material, cellulose, they actually come from the Golgi. Okay, I don't know if I asked that.
Is it Golgi Apparatus?
Golgi Apparatus, yeah, the Golgi, those vesicles full of, these vesicles, see, that end up forming the cell plate, because one vesicle touches another vesicle, right, and they kind of fuse, right, to make the new cell plate. Those vesicles came from the Golgi. I don't know if I asked about that, but I think it's spelled out here. See, it says vesicles from the Golgi make the cell plate, which matures into a new cell wall. Just in case you're wondering, where did those vesicles come from, right? To make they came from the Golgi. Here's where that term centrosome is. In case you're wondering, where's that term centrosome? Remember, that means the centrioles plus the mitotic spindle. So you see here it's called a centrosome. Even though it's technically centriole pair with mitotic spindle, it's called the centrosome. You don't need to know what an aster is, but in case you're curious what an aster is, you see the short microtubules going this way? That looks like a little star, right? An aster means star. You know, in case you're wondering. I don't think I ask about the aster at all, but aster means a star like the sun, right? Right? And it looks like a little star, right? Because of the short microtubules. Again, during anaphase, the sisters are moving apart because of motors in the kinetochore. So they are moving themselves. Then we talked about binary fission, which is very simple compared to mitosis. During binary fission, remember prokaryotes, this is how prokaryotes divide, to give 2 genetically identical prokaryotes, by the way. Prokaryotes have 1 chromosome, and it's a circularized chromosome. There's 1 spot on that chromosome, and they colored it green here. That's called the origin of replication. That's where you start copying the DNA, the chromosome, and you copy in both directions until you hit the red spot. That's the termination of replication site. Now you have 2 copies of that chromosome, 2 exact copies of the chromosome, right? 2 exact copies of that chromosome. They move apart, and then you're ready for septum formation. That basically, you know, septum formation is like, it's the cytokinesis of a prokaryote, right? But it's not pinching, is it? It's not pinching. The cell wall grows inwards. The cell wall is synthesized inwards. And what are the protein most responsible for that? FITZ, yeah, FITZ, and other FITZ proteins, but, sorry, FITZ is the main one, because you can see here, the FITZ forms a ring right at the spot where the septum's going to form, and then the septum forms. So FITZ proteins are most responsible for that septum formation. Just like, remember, microfilaments are most responsible for cleavage furrow. FITZ is most responsible for septum, right? Does that make sense? And then we talked about the eukaryotic control system with the cell cycle control system. And remember, this tells the cell how often to divide. Some cells in your body are told never to divide. They're always in G0, like your spinal cord and your heart cells and some other cells. And then some cells in your body are rarely in G0. Those things are dividing like pretty much like skin cells and those hematopoietic stem cells, which are the stem cells in your bone marrow making your blood all the time. Yeah.
Do your brain cells divide?
Brain cells can divide. You can get new growth in the brain. Exercise can actually stimulate brain growth too, believe it or not. That's kind of cool study I read. But how do your cells know when to divide, when not to divide? It's all because of the cell cycle control system, which is made up of checkpoints. The checkpoints are actually proteins. The checkpoints are actually proteins. And these proteins, their job is to tell the cell when to divide. And the checkpoints, there's 3 checkpoints, right? G1S, G2M, and late metaphase, or spindle checkpoint. What's the main checkpoint?
G1.
G1, you should know that. There might be a question. Which one's the main checkpoint? The G1S checkpoint is the main checkpoint. It's the one that basically where the cell decides to divide. And as long as nothing really goes wrong, it's going to clear the other 2 checkpoints. The other 2 checkpoints are to make sure the DNA is okay and it's not messed up, right? Does that make sense? But the G1 checkpoint is when the cell says, you know what, I should divide, you know. That's the main checkpoint. And if you don't pass the main checkpoint,
Go to G.
G0, yeah, exactly. See this G1 checkpoint, that's the main checkpoint. If you don't clear it, that's when you exit the cell cycle with G0, like in the spinal cord, right? Spinal cord cell, okay? What else? And then how do you get cancer? You get a mutation in a gene, but any gene?
No. So
No. Checkpoint gene, right? You have to mutate a gene, and those checkpoint genes are called tumor suppressor genes or proto-oncogenes. These are the genes I'm talking about. These are the genes that code for the proteins who are the checkpoint proteins. Does that make sense? You change these genes, you change the structure of that protein, it may not fold correctly. It doesn't fold correctly, doesn't function correctly. If its job is to tell the cell not to divide, now it can't do its job and the cell divides and divides and divides, and now one cell becomes 2, becomes 4, becomes 8. That's a tumor. That's cancer, right? So now you understand cancer. Yes?
How does cyclin proteins, have you heard of that?
Yeah, cyclins. Cyclins and cyclin-dependent kinases, they are basically these types of checkpoint. They are like clocks, right? As the cell progresses towards maturity, it'll build up a certain cyclin, and then the cyclin-dependent kinases will use those cyclins and then trigger another part of the cell cycle. And so it's like a clock. It's like it's like your day-night cycle.
Yeah.
The cyclins build and then dip, and then build and then dip. They're phosphorylated, they're dephosphorylated. So yeah, that's a good point. They're part of the regulation of the cell. Yeah,
But
cell
is that
cycle.
like when it goes up and down, is that like the cyclin, or are the proteins the cyclin, like it's under a group?
Well, the cyclins are proteins, and the cyclin-dependent kinases are proteins as well. And but the cyclins, those are the proteins that build or dip over time. The cyclin-dependent kinases are always around. Very good question. But yeah, that's how the biochemistry of the cell works. The cyclin-dependent kinases, cyclins, and other proteins like P53. That's a very important tumor suppressor gene. And then there are some proteins that aren't cyclins at all that are very important for the cell cycle. But cyclins are dependent in the cell cycle. Very good. Good question. Other questions before we move on? No, we're good? Okay, let's do it. That wasn't too bad. And like I told you guys before, Chapter 10, a lot of that mitosis stuff, I've already posted those videos. You can watch them over and over again and hopefully get it down. Just make sure you really understand the difference between homologous chromosomes and sister chromatids. Looks like it's still going well. Okay. Last chapter on this next exam, Chapter 11, very straightforward stuff. Let's go through it. Again, know what diploid cells are and what haploid cells means. Your haploid sperm has 23 chromosomes. The haploid egg has 23. Woo! Everything okay? Sounds like something. That was exciting. Something fell. Maybe move away from there. Yeah, move away from there. Yeah, maybe sit Something's landed on the ceiling tiles. I don't want something to fall through the roof and hit you guys. So yeah, get a very strange. I bet someone was working up there or something fell or
Something was over there or something.
Alright, I'm gonna duck under the table first.
very strange. Yeah, so hopefully, yeah, you're out of the way. Okay. Wow.
I'm gonna sit down here.
Wow, that was weird. All right. So again, the haploid sperm has 23 chromosomes. The haploid egg has 23 homologous chromosomes. They come together. That's how you get your two homologous sets of chromosomes. You have your 46 chromosomes, a mom and a dad chromosome 1, a mom and a dad chromosome 2. And remember, why are there two chromosome 1s? What do they share in common? They're not identical. What do they share in common?
The same function.
The same functions, the same types of genes. Types of genes, like hair color, eye color, right? Does that make sense? So mom and dad chromosome 7, for example. For example, I'm just giving you a random example. If you're dealing with eye color, it might be on mom and dad chromosome 7. That's where eye color gene is. And everyone knows if you're trying to study eye color, you're looking at chromosome 7. So everyone's chromosome 7 would have that gene, right? Does that make sense? That's not an accurate example, but I'm just giving you, because I don't remember exactly which chromosome houses eye color. But you get my point. Wherever the hair color gene is, for me, if it's on chromosome 11, then it's on all of your chromosome 11s have that gene as well. So the same types of genes are on chromosome 1. The same types of genes are on chromosome 2. But my hair color gene will say brown, and your hair color gene might say blonde or whatever. Does that make sense? That's what we mean by homologous chromosomes. Same types of genes, but doesn't mean the same exact information. Blonde hair is different information from brown hair, but it's the same type of information. It's color information, hair color information, right? Does that make sense? So hopefully that was clear to you guys. What else? We talked about meiosis 1 is considered reduction division. Remember that? Meiosis 1 is considered reduction division. I'll definitely ask that. And remember what that means? That means at the end of meiosis 1, you have two haploid cells. Even though they technically have 46 chromosomes, they technically have two sets of chromosomes. Because you don't have homologous sets, they're haploid. Okay, so meiosis 1, reduction division. What was meiosis 2 called?
Equation.
Equational division. Why? Because like an equation, 46 divided by 2, what do you get? 23. So the sperm and the egg have 23 chromosomes. All you did was go from 46 to 23. That's equational division. You didn't change it from haploid to something else. It went from haploid to haploid, didn't it? Mm-hmm. And meiosis 2, all you did was decrease the number of chromosomes. So that's why it's called equational division. Does that make sense? So meiosis 1 is called reduction division. Meiosis 2 is called equational division. There's only one interphase though, right?
Mm-hmm. Yes.
And it happens before meiosis 1. There is no interphase between meiosis 1 and meiosis 2. Does that make sense? There's only one interphase. It happens before meiosis 1. There is no meiosis. There's no interphase between the two meiosis. Yes?
So in meiosis, they don't have the
Metaphase.
That's a good question. The question is, in meiosis, it doesn't seem like there's a prometaphase. There's technically a prometaphase because all that means is the nucleus has gone away and those microtubules start to attach. So there has to be a prometaphase, but they just don't talk about it. Yeah, does that make sense? So some textbooks might say there's a prometaphase during.
textbook they say.
Yeah, they talk about it, but some textbooks don't talk about it because what they say is, well, it's a lot of information to know about it from mitosis. Does that make sense? So of course it's happening, but you don't really need to worry about it for meiosis. Yeah, good question. So remember, interphase happens and G1, the cell grows, S, the chromosomes get copied, so you go from 46 to 92. G2, the centrioles get replicated. And now you're ready for meiosis 1. And remember, during meiosis 1, you got prophase 1, tetrads form, crossing over happens. And, I don't know if I asked this, but the sisters are held together with cohesin, right? But this pair of sisters is held to the homologous pair of sisters with synaptonemal complex. That's what's holding the blue ones to the red ones. That's what makes sense. The sisters are held together with cohesin proteins, but this pair of sisters is held together with that pair of sisters with the synaptonemal complex, right? I don't know if I asked that, but it's good to understand. So during prophase 1, what are the main things you should know? Tetrads form, crossing over happens. Why do we do crossing over? Increase genetic variability. Perfect.
So can you explain a little bit about the chiasmata?
The chiasmata? Yeah, the chiasmata is basically during this part here where this strand infiltrates that strand and there's actual cross. When you cut this tail and that tail, when it actually forms that structure, the cross, that's what they're talking about. But you don't need to know it for the test. Yeah, chiasmata?
Basically, it can understanding like the tetrads, right?
It's basically it's how the crossing over is happening. First, you have to form that chiasmata.
Is this not Synapses, the thing that
Synapsis is the process of those tetrads coming together with the synaptonemal complex.
right, those complexes,
Snap.
are they stronger bonds than
Then the cohesin proteins? I'm not sure if they're stronger or weaker. That's a good question. Is the synaptonemal complex stronger than the cohesin proteins? Probably not. I'm not sure. It's not really a big deal as long as they hold them together.
okay.
Okay, so again, prophase 1, the important things to know, tetrads form, crossing over happens. Metaphase 1, the important thing to know,
Independent.
tetrads line up. Tetrads line up at the center of the cell with, then they line up randomly, right? And independently of one another. That's called independent assortment. So there would definitely be a question, when does independent assortment happen? Metaphase 1, right? Metaphase 1. And what's lining up at the center of the cell? Tetrads. Very good. Then during anaphase 1, what's separating from what?
Sister.
Yeah, well, there's two ways of saying it, right? Both are correct. So know this. One is the easier way of saying it. The homologs are separating, right? Homologs. What does that mean? Mom's information is separating from dad's information, isn't it? Look, both of mom's chromosome 1's heading this way, both of dad's chromosome 1's heading the other way. Homologs separate, right? Another way of saying it, which is equally correct, is this pair of sister chromatids is separating from the homologous pair of sister chromatids. Because that's what this is, right? It's this pair of sister chromatids. It's separating from the homologous pair of sister chromatids. Now someone commented, well, these aren't technically sister chromatids anymore because one of them has a little blue tail, right? And that's true. Technically, if you want to be very technical, these are called non-identical sister chromatids. Non-identical sister chromatids. They're still sister chromatids, but because of that little blue tail, it's not identical. But I don't think I go into that amount of detail on the exam. I just call them sister chromatids. But you know they're not identical, right? Because crossing over happened. Does that make sense? And then at telophase 1, you have daughter nuclei form, right? And remember, meiosis 1 is called reduction division because even though both of these cells are going to have 46 chromosomes, which is technically 2 sets of chromosomes, they don't have homologous sets of chromosomes, so it's called reduction division. And both of these cells are haploid. I may ask that. At the end of meiosis 1, are the cells diploid or haploid? And they're technically haploid. Right? At the end of meiosis 1, both cells are technically haploid. And that's why meiosis 1 is called reduction division. Because you're reducing the ploidy from diploid to haploid. That's why it's called reduction division.
But still keep the same number.
But you're keeping the same number of chromosomes because the original germ cell had 46 chromosomes. Right, the original. And now you have 46, 46. So you didn't do equational division. You didn't decrease the number of chromosomes, but you did decrease the ploidy from diploid to haploid. Why? Because now, instead of this cell having a mom and dad chromosome 1, it just has 2 of mom's chromosome 1. Now, that's not diploid. That's not diploid. You have to have a mom and a dad chromosome 1. Right? That's what I'm talking about. And that's probably the hardest thing to really grasp about this chapter, really, if you ask me. Once you understand that, you're like, oh, yeah, okay, it makes sense. Now I understand what diploid means. Diploid means not just having 2 of mom's chromosome 1 and 2 of dad's chromosome 2. You have to have a mom and a dad chromosome 1, mom and dad chromosome 2. Otherwise, you can have 10 of mom's chromosome 1 and still not be diploid, right? Does that make sense? Good. Meiosis 2 is called equational division because we're going to go from 46 to 23, right? Prophase 2, what is the DNA doing? It's forming sister chromatid pairs. And if you want to be very specific, you could say non-identical sister chromatid pairs. Metaphase 2, what's lining up? Sister chromatid pairs. What's separating from what during anaphase? The sister chromatids are separating from one another. And in telophase, daughter nuclei form. And the product of meiosis is, okay, 4 genetically non-identical cells. So what does that mean? These 2 cells, none of these 4 cells have the same information.
Not the same genome.
Not the same genome. And those cells are haploid. They have 23 chromosomes each. They are called the gametes, which are the sperm and the egg. Okay? Now, what happens if there's a failure? You see these sisters lining up? Instead of separating in half, what if they don't separate perfectly and 2 of them go one way, right? That's called what? Non-disjunction. Non-disjunction. That means the sisters don't separate correctly. Maybe 2 of them go one way. That's going to result in aneuploidy. Aneuploidy means gametes with the wrong number, or not wrong, I don't want to say wrong, the abnormal number of chromosomes, right? So instead of 23 chromosomes, you have 24 or 22. You see that's an abnormal number of chromosomes, right? Does that make sense? How did you end up with an abnormal number of chromosomes? Non-disjunction. The sister chromatids didn't separate correctly. Okay, it's considered a mistake during anaphase. Okay, and that can lead to genetic diseases, right? And the one you should know about, I don't know if I wrote it here, but it's called Trisomy 21, right? Trisomy 21. That means you have 3 chromosome 21s. You should technically have 2. And that leads to Down syndrome, right? That's the most famous example of non-disjunction aneuploidy, right? Okay, does that make sense? And these slides here, we really don't need to go into it, but I've got a question here. In terms of what the DNA is doing, right, is mitosis closer to meiosis 1 or closer to meiosis 2? In terms of what the DNA is actually doing, what do you think?
Meiosis 2.
Meiosis 2. I'll tell you why. Look here. Look at meiosis 1. Look at this is my point. Like, do tetrads form in mitosis?
No.
No. Crossing over happen in mitosis? No. Tetrads line up in mitosis?
No.
Independent assortment happen in mitosis?
No.
No. You see what I'm saying? A whole bunch of stuff happens in meiosis 1 that does not happen in mitosis. Does that make sense? But look at meiosis 2. Sister chromatid pairs form. That's actually what happens during mitosis. Sister chromatids line up. That's what's happening during mitosis. Sister chromatids separate. The sisters separate from one another. That's what happens during mitosis. So obviously, meiosis 2 is not exactly like mitosis, but it's very similar. Does that make sense? So I may ask, is mitosis more similar to meiosis 1 or meiosis 2?
Why?
Meiosis 2, because the same things are happening for the most part, right? Sister chromatids form. Sister chromatids separate. You see what I'm saying? Whereas meiosis 1, a bunch of stuff happens during meiosis 1 that mitosis doesn't involve, which is like tetrads forming, crossing over happening, independent assortment. All kinds of stuff's happening during meiosis 1 that doesn't happen during mitosis. Does that make sense? And that's that. That's the chapter. Okay, let me actually go ahead and hit stop on this. So this will be available for